The content of this video summarizes the main concept behind the John Dueber lab publication of synthetic protein scaffolds provide modular control over metabolic flux.
John Dueber's lab makes use of a natural enzymatic process known as substrate channeling. Tryptophan synthase provides an illustrative example. The crystallized enzyme complex reveals that the two components form a tunnel where the active sites of the enzymes are spaced roughly 25 angstroms apart. The tunnel is large enough to allow the passage of 4 indole reagents which act as the initial reactants. In the picture, the blue, red, and orange subsections make up for the coenzyme, while the light blue maps the substrate tunnel. Substrate channeling promotes the product production rate by preventing the loss of intermediates to diffusion to the outside cytosol, decreasing the transit times of intermediates between the enzymes by decreasing the proximity between them, and improving unfavorable equilibria and kinetics by dramatically increasing the reagent's effective concentration when bound within the constraints of the tunnel.
The artificial constructs designed by the Dueber lab resembles the natural process by possessing a tunnel with interactive sites. The construction of the enzymatic scaffold is very similar to architectural construction. The framework is first assembled by polyglycine serine residues, similar to the wood-steel frame of a building. Pathway enzymes are fused to the scaffolding by tethering orthogonal ligands to enzymes in the reciprocal protein domain partner within the scaffold. Each pathway enzyme is associated with a unique ligand-protein pair. The binding pairs are incorporated into the enzymes and scaffolding by editing the coding DNA. The design of the pathway enzymes within the synthetic tunnels are highly tunable by adjusting the orientation and number binding domains included in the scaffolds.
Intuitively, it's logical to order the enzymes in the same manner that they are needed in the overall reaction. However, the ratio of these enzymes is not as straightforward. Of the 3 required enzymes, the Dueber group held the number of the first enzyme present and experimented with the ratio of the later 2 to find the ratio which yielded the highest production rates. The last 2 pathway enzymes were adjusted between having 1 to 4 substituents in the scaffold. The mevalonate production rate was recorded for all possible combinations and compared to the production rate of the unscaffolded pathway.
The results demonstrated that mevalonate could be produced more efficiently by using a scaffolding protein. When the later enzymes were in a 1-to-1 ratio, with 2 of each in the scaffold, the production rate increased by 77-fold compared to the natural pathway. The group also experimented with the glucaric acid pathway and found similar trends. This implies that the benefits induced by scaffolding are generic and that many different pathways would most likely exhibit similar effects using this process. This research could be used to improve the production efficiency of a broad range of molecules, most notably those with therapeutic purposes. This could potentially lower the cost of Medicare, broaden the range of treatment availability, and improve the accessibility of developing medicine.